Okay, good afternoon, everyone. Can you hear me?
Yes.
Okay, great, thank you. So before we begin the class, a few things. One is I saw a couple of students already, sign up for the paper presentation. You select the papers and, sign up those which lecture you want to do the presentation. So if you haven't do so, please go through those, paper list and make your selection, okay? And also for the assignment, we have the first paper review, this unit for biomedical image segmentation that is due January 26th. So here I provide, let's see. So if you go to modules, lecture slides and videos, I kind of give you like a paper review report example, right? So actually this is one report for my student when he was taking my class when I was in UNC Charlotte. See if this will come up. Okay, so this is like basically what the report will be look like, right? So as you recall, in the second lecture, I mentioned a couple of things. When you're writing the report, right? The summary, the strengths and the weakness of the paper, and also like overall rating, and you have to justify. So the most important thing is the weakness of the paper. So that requires a lot of critical thinking. So make sure you're able to, kind of make those comments from different aspects, right? The novelty, experiment evaluation, clarity of the presentation, etc. So yeah, you can go back and see the second lecture, a few kind of guidelines. So for the report, basically you can write this a minimum of 1 page report. There's no page limit. You can go to like 2-3 pages if you want. You have more things to share, feel free to do so, but at least have 1 page report in this kind of 2-column fashion, okay? And yeah, so I provide this Word template if you prefer using Word, or if you prefer using LaTeX, here is also a template, okay? Yeah, it should be 2-column, so you can just follow this template I provided, okay? Okay, so that's it just for the paper review and the presentation sign up. Just want to give you a quick reminder, okay? So today we'll continue talking about this a couple of common imaging modalities. So last lecture, we've introduced X-ray, ultrasound image, and also computer tomography, CT. So today we'll also talk about a couple of very common imaging modalities, specifically MRI. So the magnetic resonance image, MRI, is a medical imaging procedure for making images of the internal structure of the body, okay? So just like the CT X-ray, right? You wanna see the internal structure of the body, okay? But the MRI scanner using strong magnetic fields and the radio waves to make images, right? Not like the X-ray, CT, they using ionized radiation. But for MRI, it will use strong magnetic field and the radio waves to make those images. Here, I want to show a video just to give you like an overview what MRI can do and what are the procedures if you want to take an MRI scan. Okay, so basically this MRI scanner is able to pick up the signal, right? With the proton get excited when it's go to the relaxation state, the emit energies. So the MRI scan is able to pick that energy and make the images of your different body tissues. Here is a picture show a couple of major components. So here we can see this MRI scanner. There's a big tube, right? So it's like a donut shape, which in the video you can see most of them can be closed, and some of the MRI scanner can also be open, right? One side open. And you have the one big part is the magnet, right, to generate those magnetic field. And you also have this grating coils and also radio frequency coils, which we'll discuss each of the component later. And also have this patient table. Obviously, the patient will lie on this table, then maybe they will be pushed into the tube for the scanning. So the magnet is the largest and most expensive component of the MRI scanner. So I don't know if you guys ever see an MRI scanner before. So you see it's a quite large machine, right? So a major piece is considered this kind of magnet. The strength of the magnet is measured in Tesla. So clinical magnets, generally have a field strength in the range of 0.1 to 3 Tesla. With some of the research system can be up to 9.4 Tesla for human use and 21 Tesla for animal system. So just to make some comparison, so the Earth magnetic field is only 0.5 Gauss, right? So 1 Tesla equals to 10,000 Gauss, right? You can see this MRI machine has a very strong magnetic field, right, compared to this Earth's magnetic field. And most of the clinical magnets are superconducting magnets, which require liquid helium to keep them very cool. A higher magnetic field strength increase the longitudinal magnetism because more protons align along the main axis of the magnetic field. This will increase the signal to noise ratio. So remember, we have those, 1.5 T MRI, 3T MRI, right? You can even go much stronger magnetic field, like 9.4 T MRI. So later I will show you a picture, like a comparison. What's the brain MRI look like on using a 3T MRI machine versus 9.4 T MRI machine. So the improved SNR can be used to generate the images with improved spatial resolution, right? So you can see much more or rich details of the body components, like the brain, different parts of the brain. Next component is the grating coils. They are used to spatially encode the positions and photons by varying the magnetic field. There are 3 different grating coils that is inside this MRI machine. Okay, so outside is the superconducting magnet, right? The second layer here is the grating coil. Okay, so each of these, these grating coil will produce 3 different magnetic fields that are each less strong than the main field. Okay. And the grating coil will create a variable field, XYZ, that can be increased or decreased to allow specific and different parts of the body to be scanned. So in nature, this MRI image is going to be 3D, right? A 3D volume is able to capture different slices of the body. And next component, which you see here, is the radio frequency coil. Okay, it is used to send radio frequency pulse and receive the signal back from the patient's body. There are different coils located in the MRI scanner to transmit wave into different body parts. Okay, maybe there's one placed on the head, there's maybe on the leg, and maybe one also on the abdominal region. But if you just want to scan your brain, maybe all these RF coil will send the energies there. Okay, we'll also have the computer system that is embedded in the MRI scanner. So we'll have several functions. First is to control the radio frequency and the gradient pulse, and also collecting the data, and also process and display the generated images. Okay, so the data generated by MRI scan is usually in the so-called K-space that will encode spatial and frequency space data. And then we're using computer algorithm to reconstruct, based on that data we receive, and reconstruct into the spatial space, and which will display on the computer as the image as shown here. Okay. So there's a lot of details of the physics of MRI. So if you're interested in know more in detail, so here are some of the reference I list here. You're welcome to, check these references and know more details about the physics of the MRI. And so by varying the sequence of the radio frequency pulse applied and collected, we can generate different types of images from the MRI scanner. So the most common MRI sequences used are the T1 weighted and the T2 weighted scans, right? But here we have T1 weighted and T2 weighted, right? But what does the T1 and the T2 mean? So before we explain that, we have to understand a couple of terminology. One is called the repetition time. So that is the amount of time between successive pulse sequence applied to the same slice. And also the other one is the time to echo. Okay, so it's the time between the delivery of the radio frequency pulse and the receipt of the echo signal, right? The signal reflect back. And the tissues can be characterized by two different relaxation times, T1 and T2. So T1 is the time constant which determines the rate at which excited protons return to the equilibrium. So it is a measure of the time taken for the spinning protons to realign with external magnetic field, right? So if you remember in the beginning of the video I showed you, right? So when you, the magnet will generate those external magnetic field, right? So which make your body protons align with that magnetic field. Then there's a radio frequency coil, right? To send those radio frequencies disturb that the alignment of the protons, right? So when the radio frequency was turned off, and these protons will realign with the external magnetic field and emit those energy, which will be picked up by these scanner to generate the image, right? So this T1 is a measure of the time for spinning protons realign to the external magnetic field. And T2 is the time constant which determines the rate at which excited protons reach equilibrium or go out of phase with each other. So it is a measure of the time taken for spinning protons to lose phase coherence among the nuclei spinning perpendicular to the main field. So there's a lot of I think physics on here. So you don't have to understand that right now, but basically one thing to remember is these are just two different relaxation times, tissues can be characterized, okay? So the T1 weighted image are produced by using short time of echo, right, TE, and the repetition time, TR. The contrast and brightness of the image are predominantly determined by the T1 property of the tissue, okay? So then for the T2 weighted image are produced by using longer TE and TR times, right? So T1 is short, but here we are using longer TE and TR times. So in these images, the contrast and brightness are predominantly determined by the T2 property of the tissues. So basically you will see later, I'll show a couple of examples. Different tissues will show different contrast and brightness on these T1 weighted or T2 weighted images, okay? And a third, MRI sequences is commonly used is called the fluid attenuated inverse recovery, FLAIR. So it is similar to the T2 weighted image, except that the T and the TR times are very long. So much longer, even longer than the T2, right? T and the TR times. So here is a table show what is the typical or common TR and TE times for these three different sequences, T1 weighted, T2 weighted, and FLAIR, right? So for the T1 for the TR is about 500 millisecond, TE is about 14 millisecond, right, which is short. But for the T2, we have a longer TR and TE, right? For the TR, we have 4,000, and for the TE, we have 90 millisecond. So for the flare, it will have even longer TR and TE, right? 9,000, and here we have a 114 millisecond. So let's hear a very clear explanation from a doctor actually explain what are those different modality, sequences, and what those different contrast and brightness will capture. Okay, so here maybe I can also talk about this particular example shown the T1 weighted and T2 and a flare. Okay, so for this, this is the T1 weighted image sequence. And we can see that the fat is showing bright on this T1 weighted image, right? So this outer side of fat, maybe brain marrow. And we also see the fluid, this cerebral spinal fluid, CSF, which is in the middle here, which is showing dark. And we also have the gray matter, right? So this like wiggly outside, right? So it's showing gray. And we also have the middle white matter that is showing bright white here on this T1 weighted image. So for the T2 weighted image, for the fat, it is also bright, right, outside here. But for the fluid, the CSF, which is that is the opposite as in showing the T1, right? Remember in the T1, this fluid was showing dark, but here on the T2, it is show bright, okay? And so for the gray matter, it is the outside, right? This ribbon-like is showing bright, which is also the opposite as showing the T1. On the T1, it is actually gray, right? And the white matter is white, it's kind of anatomical. But for this T2 weighted image, the white matter is showing dark, which is kind of reverse anatomical. So this T2 weighted image is good for detecting areas of pathology, right? So if there's a tumor, there will be showing some bright regions on this T2 weighted image, right? Well, although flares may be more sensitive to those pathologies. Okay, so this is a flare sequence. Okay, so it is similar to T2 weighted, except the water are suppressed, so it will appear as dark on this flare sequence, right? This fluid here. So most pathology exhibit bright signals on the flare. So we are able to, based on the flare sequence, kind of the radiologist is able to kind of detect or identify the regions that shows the pathology, like the cancer or tumor. Okay, so here is basically like a comparison of these different sequence, like T1 weighted, T2 weighted, and flare, and what different kinds of tissue will show up, in terms of contrast or brightness on these different sequences. Okay, so in this video, I'll also show MRI machine in operation. So because the MRI machine will have these grating coils, right? It will, receive these electrical pulse, and they generate a magnetic field, which is superimposed on the main magnetic field. Well, each pulse will make the coil vibrate and create loud noises. So in this video, I will show basically different MRI sequences may make different, distinctive noises. Okay. So the MRI is considered ideally, for the soft tissues, as compared to X-ray or CT, they were good for dense tissues like bones. Okay. So, MRI is commonly used for diagnosing brain tumors and also diagnosing spinal infections visualize torn ligament in the wrist, knee, and ankle, and also visualizing shoulder injuries, evaluating bone tumors, herniated disc in the spine, and also diagnosing strokes in their early stages, right, by looking at those brain MRI scans. Several comparison to CT. CT is more widely used than MRI. One is because CT is fast, and a lot of time maybe CT is enough to make some accurate diagnosis. Right, CT can take a couple of seconds, but MRI could be very slow. Right, for the sparse MRI, well, 5 to 10 minutes, but for the abdomen or brain MRI scan could take 30 to 40 minutes, okay? But one advantage of MRI is it does not have ionized radiation, right? Not like the CT or X-ray, they have the ionized radiation. And MRI is also excellent for imaging those soft tissue, have very clear contrast, but CT is preferred maybe for lung and bone imaging. So there are also contraindications to MRIs. So for example, if patients has implant devices and other metallic devices, maybe have to talk to the radiologist or physicians to see if MRI is going to be an option, because the MRI has very strong magnetic field, which will has potential influence to these kind of implants, right? Like pacemaker. And also some unstable patient. Most kind of resuscitation equipment cannot be brought into the scanning room. And also pregnancy, right? Pregnant woman, there may be some contraindications, due to some unknown effect to the fetus. And also some other factors like claustrophobic, right? Some of patient may be, claustrophobic, which, this MRI, you have a closed MRI, right? Those very tiny space, closed space, the tube. So the other, for this kind of patient, the one option is go for that open MRI, right? Which in the first video I showed, there's a one side open for those MRI. So that could be an option for this kind of patient. And we also have to care about the safety in the MRI room because of the strong magnetic field. So the MRI room has a strong magnetic field at all time, and is strictly prohibited to bring magnetic objects into MRI room. And in general, in preparation for the MRI examination, you will be required to wear, maybe headphones or earplugs to protect your hearing, right? Because of the very loud noise, which we just heard from the video. Okay. Okay, so here is a picture I previously mentioned, right? So this is brain of a volunteer that is imaged using 3T on the left, 3 Tesla of MRI machine, and on the right is the MRI of a 9.4T MRI machine, right? So, so you can clearly see that this stronger magnetic field or generate more or higher resolution, spatial resolution of the brain image, right? So it is quite clear on this image here. Okay, so because of this kind of unprecedented clear images can be show for the internal structure of the body, really help the in the medicine, it's able to make very clear diagnosis of this pathology and make very accurate or maybe early treatment for the patient. So there's a couple of Nobel Prize for MRI, right? So here a couple of physics, Nobel Prize for Physics that is contributed for the contribution to the MRI, and also chemistry and physiology or medicine. Okay, so now the technology is still advanced, right? Maybe focused on make clear image and also reduced the scanning time, right? So as I mentioned before, some of the MRI scan will take a long time to complete, right? How we are able to achieve the level of speed like doing the CT scan, which is very desirable, beneficial for patients. Okay, so here I'll show a couple of clinical use of MRI. So the first is the detecting or locating the herniated disc. Okay, so a herniated disc is a fragment or the disc nucleus that is pushed out of the annulus. So here you can see this is a graphic illustration of a herniated disc, right? So here maybe also this is a comparison which is make more clear, right? So this is the normal disc and this is the herniated disc. So by taking this kind of spinal MRI, we are able to detect, right? So this would be the point on the spine that shows herniation, okay? And we can also using MRI to detect those myocardial infarction, and also to detect those rectal tumor or colon cancer or tumor, right? So this could be, MRI scan can be used in conjunction with this colonoscopy. And it also can be used for brain tumor, detection, right? So here, I have an image that is took from the BRADS dataset, brain tumor detection segmentation dataset, right? So this is the original image, and this is the segmentation of the tumor. Okay, so next I want to talk about just briefly talk about these functional MRI. Okay. So these MRI, provides excellent spatial resolution of the head and brain autonomy, right? So we can show, we can see clearly different, tissues of the brain, right? Gray matter, white matter, etc. But the functional MRI, fMRI for short, is a technique that is able to provide alternative measure for neural activation based on the associated hemodynamic changes. So basically this fMRI is used to measure the function instead of, measure the anatomy, right, the structure. Here I want to see, what is the like a biochemical function of a specific, activities of the brain or maybe other parts. Okay, so these methods complement each other and can be used together to improve the interpretation and functional neural organization. Okay, so fMRI can measure the brain activity through the oxygen concentration in the blood flow. It relies on the fact that cerebral blood flow and the neurologic activation are coupled, right? So when one area of the brain is active, right? Maybe you are reading or, speaking, one part of brain or area is active, right? The blood flow to that area will increase, okay? So this fMRI is able to tell like which part or area of the brain is active. And so the fMRI is being used in many studies to better understand how a healthy brain works. And also it has been increasingly used to understand like how the normal function is disrupted in diseases. For example, the Alzheimer's diseases, right? So we can using fMRI to kind of see, why those normal functions is disrupted. Here, some interesting question, right? So which part location of the brain is activated when reading, and which part of the location of the brain is activated when listening to music, right? So here, so in this article, they provide this kind of study using functional MRI, trying to map, right, which region is active when when the subject is perform like reading, listening, or maybe both together. Okay, so as you can see from this sub-image A here, right? So this red region is the legend indicating this listening comprehension. So these red regions are active in the brain. And when it comes to reading, the green one shows the active area of your brain. Okay, so the white one is show the region, or maybe some overlap regions when you're doing the both listening and reading comprehension. Okay, yeah, on the right, this is just the area indicating, you know, each individual task was performed either reading or listening. Okay, so yeah, if you're interested in more details, you can go through this article. And this is a typical fMRI setting, right? So the patient or subject, you know, you have the cameras and the coil illuminator fixation stimulus, right? So you know, the stimulus will give you, maybe ask the patient or subject to see different objects, right? Here are some maybe different objects, or maybe listening to different piece of music, and then trying to using an fMRI to measure like which part or area of your brain is active, you know, when the subject is respond to this different stimulus. Okay. Next is the nuclear medicine imaging. It may seem intimidating because of the word nuclear, right? So it's kind of scary, right? You're using some radioactive, you know, component, you know, to generate image. But it's actually is not very dangerous, very safe. So the nuclear medicine is a medical specialties that are using radioactive tracers, right, to access body functions and to diagnosis and treat diseases. Okay, so and we use specific design cameras to track those paths of these radioactive tracers, right? So basically, we'll, you know, injecting these radioactive tracers to the human body, then using cameras to detect the path of these radioactive tracers to generate those images. So the two common nuclear medicine image modality is, one is the single photon emission computed tomography. So that's called the SPECT. And sometimes you will see just SPET, so they may omit computed, so just SPECT. The other one is the positron emission tomography, PET, PET scan. Okay, some of the basics of the SPECT imaging. So the SPECT imaging will provide three-dimensional images of the distribution of the radioactive tracer that has been introduced to the body, right? So this tracer will allow the doctors to see the blood flow to the tissues and organs, right? How maybe the dynamics of your you know, metabolic system works. Okay, and the SPECT imager will have gamma cameras detectors, that is to detect the gamma ray emission from the tracer. So basically the tracer will emit gamma rays, and the specific design of the camera will able to pick up those gamma rays and, you know, make those images like this. Okay. So the cameras are mounted on the rotating gantry, so it will allow the detectors to move around the patient, so it is able to generate those kind of 3D imaging. Okay. So the tracer is radioactive, right? So it will emit a gamma ray. It doesn't, mean this is dangerous, right? Usually these radioactive tracer will injecting very, very tiny amount, okay? And also because of the radioactive chemicals have short half-lives, so they break down quickly and eventually will be removed by the kidney, okay? So it's fairly, it's pretty safe. PET imaging is similar, but the differences is, make different radio tracers being used. Okay, so the SPECT scan measure the gamma rays, and the PET is using the decay of the radio tracer, and it will produce small particles called a positron. Okay, so a positron is a particle with roughly the same mass as an electron, but with opposite charge. Okay, so these react with electrons in the body, and when these two particles combine, and they annihilate each other, and this procedure will generate a small amount of energy in the form of 2 photons. Okay, so basically 2 gamma rays into different opposite directions. And these PET scanner is able to pick up those energies and create these image of your internal organs, okay? So these nuclear imagings like PET, SPECT can be combined with MRI or CT, right? So to provide rich information or extra dimension of information for diagnosis, right? So here is the PET scan, the CT scan combined together, so you can, provide better details for the physicians or radiologists to do diagnosis the pathology. And also here on the right is the MRI with PET combined together. And here's an example. If you want to detect the tumor, right? So if we're just using the CT, as you can see, the tumor margin is poorly defined on the CT scan. But if you're also using the PET scan, right, you combine with CT, then the tumor region can be easily, detected. So that's the advantage of using this kind of hybrid or combined, scans. Okay, so if you are interested in learn more about this kind of nuclear medicine, so there's a very good lecture on YouTube, so you can introduce some of the basics and different, diagnosis different diseases using this kind of nuclear image like PET, SPECT. So you can, watch this video. Okay. So for this kind of combined or fused imaging, before you have this kind of like a separate machine, right? You first do the PET scan, then you go through this MRI machine to generate another scan. Maybe then you do some registration and fuse them together, right? So, but now here we are able to advance the engineering and have this kind of like a combo, right? It's just a single machine that you can take MRI or PET scans together. Just within the single machine, so you don't have to go through this kind of 2 machines in series. Okay. Okay, so those are some of the very common imaging modalities we've briefly talked about. And also here I want to give you some of the free softwares that can be used for medical image analysis. At least if you want to visualize some of the images like CT, X-ray, or MRI. So these are some of the options free software you can use to visualize those medical images. So next, I will actually give you a demo, using this ITK Snap software to kind of visualize CT scans and MRI scans. Okay. Before that, just a quick, mention of the medical image format. right? So medical image format are different than the, like the, conventional, image format, like a natural image, like JPEG, MPEG, PNG, MPEG is for the video. So for the medical imaging, it has some, dedicated, formats for this DCOM and this NIFTI, analyze, right? So one data format we're gonna look at is this NIFTI, which I'll later show you. And this is a file format, created at NIH, National Institute of Health, with the intent to create a format for neuroimaging, maintaining the advantage of analyzed format, which is also another, image format, but, kind of address some of the weakness of these, format, analyzed format. Okay. Okay, so let's, first, so this software name is called ITK Snap. Okay, so actually you can go to this website. So are you able to see my web browser?
Yes, Dr. Chen.
Okay. Yeah, so you see here, this software is able to install on different operation systems, right? You can install that under Windows. You can install that in the Linux or maybe install in the Mac. I'm using a Mac computer, so I actually have this opened. Okay. Okay. Yeah. Actually, before I open an image file, I want to quickly kind of recap what we learned from last lecture, right? So we know for the CT, right, which is a 3D volume, right? So we have these sagittal view, coronal view, and axial view, right? So these are the three different views or planes, and they show like the images under different views, okay? And also under each view, you have different slices, right? Because the CT scan is a 3D volume, right? You have different slices of your body, okay? And also on the, here, just some of the, kind of locations or directions indicated in this kind of anatomical space, right? We have right and left, okay, as shown in this image. And we also have these anterior or ventricle, so which is showing the front, right? A indicating the anterior, okay? And also we have these posterior or dorsal, okay? So which is like the back of the body. And also we have inferior, okay? So like pointing towards the bottom part of the body, like the leg, and the superior is pointing towards the upper, right? Your hand is like the upper extremity, right? Okay. So let's set this aside. Okay, so this is the interface of this ITK Snap, okay? So when you open that software, this is what will look like, okay? On the bottom, you will see there's like a button here, open image, open workplace. You can also click here, like you can open main image here. So let's just click open image, right? So this pop-up will, ask you like where your image file is located. Okay, so I already get an example image. Okay, so here I will show two examples. One is CT for the kidney tumor. The other is breast, so that is for brain tumor. That's the MRI. So one CT, one MRI. Okay, so let's first look at CT. So under this example, I have two folders. So one is image, so that's the raw data. And also because this task is for kidney tumor segmentation, right? So I also have a file that is the label, right, indicating where's the tumor, where's the kidney tumor, and where's the normal region. Okay, so let's first open this the raw data, which is in this NIGZ, which is the NIFTI format, which we just talked about. So we'll load the data. Okay, so this is, let me just blow this up. So this is the CT scan look like on the abdomen region, right? Because we are examining the kidney, right? So the stomach region here, okay? So one thing you'll notice, right? We have three views, right? We talk about here, right? We have sagittal, coronal view, and axial view, okay? Okay, so actually I want to ask you a question. So you see these three views, right? Which one is axial view?
Top left.
Yes, this one, right? This is the axial view. How about this?
Like if you see from the side.
The
Yes.
sagittal.
Yes, that's the sagittal, and this is the coronal view. Okay, so let's see why is that. Okay, maybe if, yeah. So this one, you know, based on the common sense, right? This is feels like you're looking on the side, right? The sagittal view is right. You look at the left and right, right? The plane here, right? Corona is like front and back, and this axial is like top and bottom. Okay. But we can also determine by these, I don't know if you can see these yellow letters here, like S, I, here is A and a P, right? A is anterior, right? Anterior is like the front, right? P is posterior, right? Here, look at, I'm doing this on the side-by-side comparison, right? So hopefully you can see this small region on the right. So posterior, right? So we know this is front and back, right? So superior and inferior, right? This is the top and bottom, right? Which means we are looking on the side, right? The sagittal view of the person, right? So here again, this is A, right? The front and back and left and right, right? So here we are looking at the top to bottom view, right? So that's the axial view. Okay. So now you understand like which view we are, which view is to which, right? Which plane we are, you know, examining, you know, we are taking this slice. And let's maybe here focus on just one view, view here, the top left, which is axial view. Okay. If you left click your mouse here, okay, and you scroll your mouse, right, up and down, okay, you can see this image, this view change, right? So that is showing you different slices in this view, okay? Remember, the CT is 3D volumes, right? We take multiple scans, right, of the human body, right? Scan line by line doing a scan, right? So we take different slices of your body, right? So that's why you'll see, you know, different, different, slices in under this view. Okay. I don't know if you notice, right? There's this line like kind of moving up and down, right? When this axial view change, right? See that? And also on the corona view, right? You see that blue line moving up and down, right? So that makes sense, right? So here, I'm actually, this is the plane, the slice I'm taking, right? This slice corresponding to the axial view here, right? If I'm moving down like here, right? This is a slice I take from here, which I viewed on this sagittal view, and this is the corresponding axial view. Okay, so so you can actually visualize like different slices in one view. Okay, so when you click on this, on the sagittal view here, you can also scroll your mouse. Okay, then you can see the different slices of your sagittal view, right? You can see this line move, move left and right, right? Okay. Right, you are able to examine different slices of the body. Okay. This makes sense, right? So let's say here, this appears to be the human spine, right? Imagine this patient is lay flat on this, table, right? Performing the scan, right? This is the spine, right? The back of a person, right? So it's corresponding here, right? Okay, you can clearly see the spine. And this is the coronal view, right? So like the front and back. Okay, you can also scroll down and see how each slices was taken under this view, right? So if you look at these, how this move on the sagittal view, right? This is like your skin of your stomach, right? This is your back, right? You can see how it is kind of able to examine different slices. Okay, right, so let's see here, I'm able to see this kind of, the bones here, right? Probably connecting the leg. So you also see that, right? I'm taking this slice on the coronal view, right? So you can see here, okay? As you keep moving towards the very end of your back, right? You basically just seeing some of the fat tissues here, okay? So that's how you can, kind of move around and, examine, different slices from different views, okay? And now what we want to do is let's, open the segmentation, right? So on the segmentation, you open segmentation, and you can, because we have the labels, right? We know this is the ground truth label that indicating the tumor or non-tumor region, okay? And when you load this, you want to update here on the left. So this, you want to do the update. Okay, so you will see this 3D, ground truth of the kidney. So the red one is the normal tissue of the kidney, and the green is the tumor. Okay, the green is the tumor. Okay, so now you can see how different tumors will present on different slices, right, on the different views, right? So if I place here, okay, so imagine like on the side, right, I'm cutting here, right? I'm taking this slice, right? So on the sagittal view, right? So you also see this tumor. I mean, this is the normal, right? Red is normal, showing on the sagittal view, okay? But for the coronal view here, we are actually able to see the tumor. Why is that? Maybe this one we have occlusions, right? The tumor may be blocked by this healthy region, maybe on behind it. So we are not able to see from this slice, but we are able to visualize that on this coronal view. Okay. So so any questions when you're playing with this? Okay, so this is the CT. Now let's open, let's look at MRI. Okay, so let's look at the brain image. So if you look at the name, right? So this is the like a patient numbering. I know this is maybe a little bit small. So actually you can see like T2, T1CE is actually another sequence, other modified based on the T1. But here, well, let's just look at T2, T1, and the flare, right? These sequences common sequences we mentioned. Okay, let's load them. Okay, I want to add another. So this is T2. Okay, let's add T1. Okay, so you see there's another one add here. Okay, I want to do comparison. So I want to preload these three different sequences. So let's add another one, add another image. Now this time we'll load flare. Okay. Okay, so if you click here, right here indicating this is T2, this is T1, and this is flare. Okay, so remember when we have some different characteristics of these different sequence, right? For the T1, so the fluid. This is T2. This is T2, not T1. Right, this is T2. Let's look at T1 first. Okay, so T1 is the fat will be bright, right? Gray matter will be gray. This is like anatomical, right? So gray is gray, white is white, right? White matter is white, and the fluid, right? CFD, CSF. This is the fluid here. We're showing the dark. Okay, so for the T2, it's kind of reverse anatomical, right? So the white will be gray, right? So we have this fat is going to still be bright, but for the white matter will be dark, but for the gray matter will be bright, right? So which will show like this ribbon-like. Okay. Also, the fluid will also show as bright. Right, so in the middle, you see that, that's bright. Okay, actually, I don't know if you notice, this is actually a tumor region right here. As we mentioned, right, so the T2 is able to kind of, have the contrast to show the pathology, right? So actually there's a tumor right here. Okay, let's look at flare, right? So here you'll also see, the fluid will be shown on the dark, on the flare. But for some, the pathology will show on the bright, right? So this is the tumor. So let's, again, do some scrolling down, right? To look at different slices. Okay, I'm looking at the flare sequence, right? I'm looking at different slices of the brain. Okay. Right, so this is the axial view of the brain, right? So I'm scrolling down to, check different, slices of the brain, okay? So you can do this on the other view as well, right? You can, check different slices here as well. Okay, then let's, introduce or load the segmentation map. Okay, so I'm going to load the label. Okay. So these different, colors, you see the red, yellow, green, indicating different type of, tumor regions. If I unload segmentation, right, just to give you some idea, right? So as I mentioned, right, these pathology is able to show bright color on this flare or T2. this is T1, T2, right? You see this bright region, right? If you look at the flare, this is maybe more clear here, right? If we add this segmentation, right? So actually this is indeed kind of highlighting these region, right? So this is the tumor core region, okay? So yeah, so if you kind of, taking different slices, you will see, different segmentations maps or lay on each of slices, right? And why is some of the slices don't have tumor? Anyone?
Because maybe because it is not looking at the plane where the tumor was present.
Yes. So remember, everything is in 3D space, right? So each tumor region has a volume, right? It does not occupy the entire brain, right? And we are looking at individual slice, right? Maybe that slice does not contain the tumor. It's all healthy tissues, okay? Okay, so this is, just some very basic, you can using this software, play with it and to see, how different slides look like, and different, for MRI in particular, you are able to, switch different sequences, and, by comparing, different sequences, maybe that will help you kind of trying to identify some of the pathologies, right? For specific, the tumor regions, okay? And this is like the entire, like a 3D view of the tumor, okay? So this software can do just more than visualization. If you are, creating a data set, right? Obviously, you need some expert knowledge, right? You have to know exactly where the tumor is, right? If you are creating or annotating a new data set, right, you can using this software to annotate, right? Actually locate, make some of the annotations, right? Where this is the tumor region, and do some of the segmentation with this, software, okay? But here, mostly we're just using this software for visualization. And later when we talk about actual, medical image computing, we'll just develop AI algorithm to automatize the process, right? So we are using AI or machine learning technique to automate this kind of detection process, okay? Okay, so yeah, so that's all for today. I think after the class, you can download this software, and I've shared this data. Let's see. So I've already upload the data, which I just demonstrated here. So you can download these data, right? Have the CT, MRI, download this software and play with yourself, trying to understand like these different views, and how they are able to provide a different view perspectives of the the body, right? If you still I know sometimes it could be difficult to imagine, right, this 3D space, but maybe you can make some practice, right? Like think about you are take a mango or apple or something, right? And maybe you can cut into slices, right? So this will kind of practice like what is this sagittal view, coronal view, axial view look like, right? So this maybe help you strengthen those kind of orientations in your head when you look at these different scans, okay? For the next week, we're starting to talk about deep learning for medical image computing, and we'll first do some crash course on deep learning. Some of you don't have any background in deep learning or machine learning, so I will spend a couple of classes or lectures just to quickly help you understand what are the deep learning techniques and how they are used for medical image processing, okay? Okay, any questions?
Professor,
Yeah,
I can't access the Excel spreadsheets for the paper.
yes, yeah. Yeah, thank you for bringing this up. Actually, there's like 2 or 3 students send me email about this. Yeah, I think for some reason, like when I use your email address, it says you are outside of the university. I think the OneDrive, they don't allow people outside the university. They don't have those access, and like anyone can using the link. They only allow like university people to access to the document. But can you send me your email? I figure out a way to share that outside, so I can share that with you. Or if you could, you can tell me like which paper you want to select and which date. Obviously, some of the papers already been taken. If that's the case, I can tell you. But I think the easy way will, can you send me your email address? Then I'll try to share that with you. Okay.
Okay, thank you.
Okay. Okay. All right. Sounds good. Okay. All right. Thank you, everyone. I'll talk to you next week. All right. Everyone have a nice weekend. All right. Bye.